Okay, let's look more specifically then at how this messenger RNA is going to take this code out to a ribosome. So we're going to go back into the nucleus here, and so to the left of our dotted line here is going to be the nucleus, and over here to the right is going to be the cytoplasm. This represents what we call the nuclear membrane. It's just that keeps the DNA nice and compact, compartmentalized into the nucleus. Well, what's going to happen is first of all, we're going to need to look at the structure of RNA just briefly. And what we'll find with RNA, it's a single-strand molecule rather than a double-strand molecule, such as DNA was, so therefore it's smaller. And if we look at our bases in RNA, we're going to find that adenine is present again, cytosine will be present again, guanine will be present again. There's going to be a new base. It's going to be called uracil, which we'll abbreviate as U. And so RNA will have uracil, but it won't have thymine. So uracil, sometimes we say, replaces thymine. But other than that, we have the same four bases. Now what will happen is that DNA will, what we say, unzip sometimes. And so what that means is that we'll see a break in these chemical bonds that are holding our A's and T's and C's and G's together. So the two strands of DNA will unzip or come apart, just temporarily. What will happen now is RNA is going to form along one of the two strands. And for this example, let's just use the left strand. It's not always the left strand, but let's use that as an example here. So what we're going to have here is RNA, we're going to form it along this left strand. Now, our base pairing rules are essentially the same, except that we don't have thymine, we have uracil. So what we're going to do then is RNA will form a uracil, will bond with the adenine, guanine with the cytosine, again, keeping our base pairing rules all the way through. And what we have now done is we have formed a small piece of RNA. And we're using a different color of that just to try to emphasize the difference between which ones are actually RNA and which ones are DNA. Sometimes students will ask, where do these come from? And these particular bases, they're found in your cells. So you have those. They're ready to be put together in some sort of meaningful way or directions. So now we're going to look at how the messenger RNA can actually accomplish getting this genetic code from the nucleus out to a ribosome. So we have formed our messenger RNA, illustrated in our different color. And what's going to happen now is the chemical bonds that are keeping the RNA attached to the DNA are going to break. And the RNA molecule then is small enough that it can get through the pores in the nuclear membrane, and it's going to come out and actually attach to a ribosome. So our RNA attaches to a ribosome. Now, why this might not look like very much to you and me, this is actually a set of directions that tell the ribosomes exactly what protein to make. And that, of course, is based on the DNA which an organism inherited from their parents or parent.
Vizi online texts are a new kind of textbook, built with the help of great subject matter experts. These new learning resources have already helped thousands of students succeed. Our original content uses animations, simulations, video and graphics, self-assessments, exercises, and review materials to engage students in their study. Our approach is proven to improve learning outcomes, and that's why Vizi exists, to transform lives by making education more engaging.